Thank you very much, Lloyd, and apologies for interrupting your gastronomic feast down there. I've had a long-standing interest in Alopecia Areata. I did my PhD in Alopecia Areata back in the early 90s, and I have always been wondering why some of our target antibody-mediated reactive proteins on the hair follicle have never appeared on any GWAS or any other genetic screens. Well, I was wondering, what really is the connection between the epitopes in the hair follicle that autoantibodies react to and the actual disease itself. And the last few years have been kind of increasing a lot of association between the skin and the gut, in a sense the skin on the inside, and the brain axis between these, and I think this has some connectivity with Alopecia Areata overall. There is some evidence from the literature that there may be association between Alopecia Areata and celiac disease. We know that celiac disease can have skin phenotypes, particularly the case of dermatitis herpetiformis, and may also have others. There's also been some co-occurrence of the two disorders, not very high, but there may be some subclinical forms of gluten sensitivity that we don't really pick up. And we have known and been very interested in the GWAS reported links between some loci that are in common between Alopecia areata and celiac disease, which may appear strange given the gluten link with celiac disease being a plant-based antigen. I have been looking at trichohyalin for a long time. It's one of the first differentiation-associated antigens to appear in early Anagen hair follicles, where the trichohyalin is on pretty much the outside of the hair bulb at that particular point. And I wanted then to see whether there could be any association between the processing of the trichohyalin protein and the processing of the gliadin proteins in terms of two particular important enzymes, the PAD enzymes and the transglutaminase enzymes, and that will be the focus really of the presentation. So the question asked was, do human hair follicles express cross-reactive epitopes between alpha-gliadin, which is immunogenic in celiac disease, and if they do, where in the hair follicle could such cross-reactive epitopes reside? And also, do patients circulating IgG, serum antibodies in Alopecia areata, react with alpha-gliadin? And we chose a particular peptide of alpha-gliadin that's associated with the immune response in celiac disease. So this was based on an immunohistochemistry approach using normal human scalp skin and an antibody to trichohyalin, AE15, that only reacts with the trichohyalin granules throughout the inner root sheath, and then followed on with an immunoblotting or dot blotting approach where we have taken this immunogenic gliadin peptide, dot blotted it on membrane, and then reacted it with Alopecia areata serum to see if we had any binding. So the results were pretty much as follows, but the background has that circulating antibodies in human alopecia, mouse Alopecia areata-like hair loss, equine AA-like hair loss, and even canine hair loss all have antibody reactivity to trichohyalin, which is this 220 kilodalton protein that you see up here, immunosuppressed in this region of the inner root sheath. And we also were able to immunoprecipitate this trichohyalin peptide from a crude extract of hair follicles with the both with the monoclonal antibody and also with Alopecia areata serum, suggesting that we do indeed have circulating antibodies to trichohyalin. We followed on by taking this immunoprecipitated and then tried to do mass spec on the proteins within the immunoprecipitate and found that a predominant peptide to come down was trichohyalin. So if you react to antibody from Alopecia areata with the human hair follicle, you find often patterns like this. And if you co-localize with the antibody to trichohyalin, you can see several regions of co-expression, particularly in the differentiating Henle's layer, the outer part of the inner root sheath, as you can see here in yellow. And if you look at this at higher magnification, you can see some quite delicate decoration of different parts of the inner root sheath co-localizing the antibody to trichohyalin and the antibody in Alopecia areata serum. So we really do believe that trichohyalin is involved somehow. I know Kevin McAlvey and others have shown. And if you look at the location of this is really quite external in the hair bulb. So it's not locked inside the hair follicle as some people may think inner root sheath proteins are. So the connection then with the alpha-gliadin peptide is as follows. We have an antibody to alpha-gliadin, you can see in green here. an antibody to trichohyline, seen in red here, and then the merged picture here where you see co-localization. And there's several parts of the inner root sheath that are expressing epitopes that are shared on both alpha-gliadin and human trichohyline. And if you look at this at higher magnification, you can see a very clear co-localization, but not total, between these two peptides. And it may be associations between the maturation of this trichohyline during different phases of the hair follicle growth. Higher power, you can see some areas of co-localization. Here we have specific decoration of particular inner root sheath cells, some of the very granular trichohyline here, and then the co-localization. It's partial overlap between trichohyline and alpha-gliadin, but there is definitely some shared epitope parent here. And if you look very closely, you can see the alpha-gliadin antibody decorating granules within the inner root sheath, and these can be co-localized also with trichohyline. It's not a complete overlap. You can see very clearly here that the inner root sheath trichohyline is expressed over a broader region of the inner root sheath during maturation. This is on the upper part of the follicle, the lower part of the follicle. But if you look where the white asterisk is, you have co-localization. But where the pink or purple asterisk is, you do not, so indicating that the gliadin epitope is expressed at a particular point in the maturation of trichohyline. I should mention at this particular point that one of the key connectors between alpha-gliadin and trichohyline is the fact that we have conversion of arginine to citrulline. And this citrullination of the peptide appears to make it more immunogenic in celiac disease because we have a change in the protein conformation. And we have lots of antibodies in these diseases, including rheumatoid arthritis, multiple sclerosis, that show antibodies against the enzymes that do these biotransformations, the citrullination via the pads, but also the linkages between glutamine and lysine in the context of transamine, the enzyme that I've suddenly forgot the name of, transglutaminases, yes. So there is a common pattern of biotransformation of these molecules that are occurring both in gliadin and in trichohyline. And then finally, we wanted to see by dot plotting whether patient sera in Alopecia Areata could bind a very short stretch of alpha-gliadin, only a few peptides, and you can see that there is indeed the activity. These are preliminary results, but it does suggest that there could be antigen mimicry across a whole range of different proteins, including plant-based proteins, and again indicating a link with diet in some of our skin conditions. So by way of summary, I would like to say that Alopecia Areata IgG appear to target inner root sheath antigens, some of which are biotransformed antigens on the basis of differentiation. There's also a key crossover across all mammals in the context of trichohyline, which I think is relevant. You also find that a very small strip of alpha-gliadin antibodies to a small strip of gliadin can bind both the inner root sheath and also the medulla. You find there's a granular pattern in this expression which is indicative of the structure of particular proteins within the inner root sheath. And then finally, citrullination is very important. It's more immunogenic when you get to citrullination. Also, citrullination antigens are more easily bound by the MHC class 1 and 2 molecules, which may be relevant here. And then finally, we need to think of the enzymes and whether they are also immunogenic in the context of these diseases. I'd like to thank you very much for your attention.